And it didn't tell me it was recording. I guess it must be though. Yeah, it is. Okay, let's share the screen and get right to it. All right. Sorry, guys. Just doing a little bit. Just getting all set up. Here we go. Okay. So, oops. Yeah. So we kind of left off talking about the inflammation response. What would be triggered if you had an injury, like stepping on a nail, and how all the different agents come in to attack that, not only the wound, but the microbes that have been introduced via the causing agent of that wound. And that's where we left off. Okay, so diapedesis and chemotaxis. Diapedesis is the migration of cells out of blood vessels into the tissue. Okay, so like I said, your blood vessels are a bunch of cells, and they have gaps in between the cells in between the blood vessel cells for your immune cells to extravasate out. And when they cross that barrier outside of the circulatory system, then that's called diapedesis. Once they're there, they will be guided by certain chemicals, and that's called chemotaxis. So chemical, taxis, to move. So once these immune cells are in the blood, they will seek this scent, if you will, not a real scent, but a chemical signal to then migrate out of the blood vessel and toward that area. And those chemotaxis factors are released by other cells in the area. It's kind of like sending up the signal, right? Like, hey, we need backup here. Okay. And that's how these cells communicate. They don't have walkie-talkies, right? They don't have voices. They have chemical signals that they send out and secrete that draw the cells toward them. So fever, what is fever? Is that first line, second line, or third line? Second. So a fever, anybody else think it's something different? No, okay, second. A fever, yeah, it's caused by the introduction of foreign agents. And it is a response that is then initiated by these circulating molecules called pyrogens, which elevate the body temperature as a way to kill off these germs. You can have exogenous pyrogens. So those are the pyrogens coming from the microbes. For instance, endotoxins that they release. Those endotoxins are meant to damage the host, but it actually triggers the immune response in the form of fever. Endogenous pyrogens are going to be what the body produces to cause fever. Okay, so the monocytes. And remember, monocytes, those are what? Do they come from myeloid or leukoid cells? Lymphoid cells. Monocytes on that lineage.
Are these proteins or what?
The pyrogens?
Yeah.
Yeah, they can be, yeah, mostly they're proteins.
Is it just like a general term for something that raises the body temperature?
Yes, it's a general term. So pyrogens can be exogenous, meaning they come from the microbes, and that triggers an fever, or they can be endogenous, meaning that the immune cells release them. Okay, so some pyrogens are going to be called interleukins or tumor necrosis factors, IL or TNF for short. And yes, these are like little proteins that are out there triggering a fever. And the IL and the TNF also trigger inflammation. They also are signals for immune cells to communicate, which we'll get into in the next chapter. So some of them have double uses, not just to induce fever, but to communicate that something's wrong to other immune cells to then come and take out the germ. Development of monocytes and macrophages. So remember, the myeloid cell can differentiate into a number of different types of precursors for these immune cells and red blood cells and platelets. The monocyte lineage goes into macrophages and dendritic cells, right? And so what happens is these macrophage and dendritic cells survey the area, the body. They just travel around looking for bad guys, okay? And in some cases, once they've migrated around, they may stick a while in a certain organ or tissue and just be there, poised and ready, okay? These are the ones that are part of the second response, right? The second line of defense. And so they're going to be there waiting for something to happen. And then once they do, they initiate the third line of defense. The receptors that these macrophages use are called TLRs, or toll-like receptors, okay? And they're going to use those toll-like receptors to recognize a foreign antigen, okay? A foreign agent, something that's not part of the body. And once they realize that, it brings those TLRs together. And once those are together in proximity, that elicits a signal to the nucleus to then start churning out these pyrogens, okay, and these cytokines and chemokines to initiate the immune response. And we'll talk about the different, so all of these immune cells have lots of different receptors, but we'll just highlight the few that are specific and major important players in the immune response. But for the phagocytes, remember that it's toll-like receptors that they use to initiate and detect the antigen. Later, we'll get into antigen presentation, and those are going to use different receptors. So when an infection has been detected, the white blood cells are going to produce interferon alpha, interferon beta, and interferon gamma. And these are going to be primarily in response to viruses, to viral infection. But it can be triggered in response to other things as well. But these are the major players for viral infection. And it's actually used as a therapy in patients that have viral infections. They might be treated with something like interferon gamma, for instance, as a way to kind of boost that response. If the body isn't producing enough, they'll treat the patient exogenously with interferon gamma. And these are going to then bind to the surface of that virus, and then trigger antibodies to come and cover it up and surround it and take it out. Okay. Any questions on that? Okay. As they come, feel free to interrupt me, because I know this is a lot of information. So if something's unclear, feel free to jump in.
I have a question.
Yep.
Just to clarify, when you're saying white blood cells, are you talking about myoblasts and monoblasts? Like the cells that come from those, or?
No, primarily when we talk about the the white blood cells. Let's see if I don't know if I have that. I don't know if I have that up, but yeah, when we're talking about white blood cells, it's going to be all of the immune cells primarily. Okay, so we have red blood cells, which are the erythrocytes, non-immune, and then we have the white blood cells.
Okay, so basically everything except for the red blood cells in the plasma.
Yeah.
Okay.
Yep. Thank you for that clarification. So, but again, the whole immune response is just a large series of signaling, okay? One cell detects something, it sends out a signal. That signal is recognized by another cell that then sends out signals, right? And so it's this progressive display of signaling and secreting things and releasing things to communicate with the body that something is inside it that needs to be taken out. So here we have a virus, right? It binds to the cell of its host. It then unpackages and releases its nucleic acid, and we're going to talk about this next week when we get into viruses. And it starts assembling, it starts reproducing its genomic content, and then translating its new nascent proteins that will encapsulate that genomic material again and then release it. But once that happens in that cell, the cell recognizes that it's infected, and it sends a message when the virus is inside the cell for it to start transcribing and translating interferon. Okay, and then that interferon is released, and then that is detected by a neighboring cell. And so by the time the new virus that has been replicated in that previously infected cell has released itself and is going to attack another cell, it's already got the memo. It has the message from that cell ahead of time. And now it is poised to start releasing these antiviral agents, okay? This is part of the innate immune response, not the adapted. We'll get into that later, right? This is the body saying, oh my gosh, we got something that's infecting us. We got to send that and communicate that with the rest of the body so that they're poised and ready so we don't have a systemic infection, okay? So the interferon is a warning signal to uninfected cells what's about to happen. Well, that's it. That's all for that chapter. Any questions on that? Okay. Let us begin with the new chapter, the adaptive immune response, okay? Now, this is a little bit more complex. The adaptive immune response requires a higher level of sophistication in a certain way because these cells have to be selective to particular antigens, the things that are triggered as being foreign. And they have to reproduce a bunch of cells that will now be ready in case that antigen comes back. And so they have to have memory as well. They have to be selective, and they have to have a memory when the antigens are reintroduced. And so that requires a different process. And that's what we'll get into today. So for this chapter, it's going to be looking at antigens, right? Antigens are just molecules that trigger an immune response. They can be microbes. They can be molecules on our own body. For instance, with blood, we all have our specific blood type. And if you get the wrong blood type transfused into you, that's not a good thing, right? If you get an organ from somebody else, unless you get immunosuppressant drugs, your body's going to try to attack it because it has antigens on it that are considered foreign, because it is, right? Someone else's blood is technically foreign to you. So antigens aren't just microbial in nature. They can also be from cells of other species or within our own species. We'll talk about that a little bit more. We'll talk about the lymphocytes, right? The T cells, the B cells, and the natural killer cells, and how they are these. And, you know, when we talk about immunity, we can't help but kind of anthropomorphize them and talk about them in militaristic terms, because I don't know, that's kind of the way it works in our minds. And so it's always this warfare going on, and lymphocytes are skilled troops of immunity. And so that's just a part of the colloquial way of referring to these things. And we'll get into the different types and how they differentiate to be specific to certain antigens, and how we then, as a result, develop this vast repertoire of immune cells poised to go after something specifically. Preparing for the battle. See, again, the battle, right? All these combat terms. We'll talk about how the antigen is detected by the macrophage or the dendritic cell, and then processed, and how that is then communicated to the lymphocytes, the T cells, and the B cells to start ramping up adaptive immunity. T cells can be helpers or killers, and we'll talk about the differences, what makes them helper versus what makes them killers. And then we'll get into the actual process of creating antibodies, the structure of an antibody, how they're produced, and how they work, if we can get to it in time at the end. So that brings us to the third line of defense. We've discussed the first line, like skin, tears, crying. We've talked about the second line, the monocytes and things like that. And now we're going to talk about the third line, adaptive/acquired immunity, the B cells, the T cells. If you are immunocompetent, then that means you have a wide variety of B and T cells that are ready to take out foreign substances. If you're immunosuppressed or immunodeficient or immunocompromised, then that means something's going on with your immune system to the point that it's not sufficient on its own to heal itself. The antigen, like I said, is the molecule that stimulates a response from these T and B cells. And that antigen can be from a microbe. It can be from someone else's blood that's a different blood type. It can be from, the pollen and flowers that a lot of us deal with. It doesn't have to just be a microbe. It could be cancerous cells. Okay, we talked about cancer a little bit when we were talking about Henrietta Lacks, and I said that what makes a cancer cell is that it's mutated. And all cells, what they do is they take a little bit of their proteins that they've translated off of their genome, and they spit them out on the outside of their cell membrane. And so your immune cells come over and say, oh yeah, I recognize that little fragment. That's part of my body's genome, so that's good. And then it moves on. But with cancer cells, they have those mutated sequences reflected in the proteins that they translate. And so that's an antigen, and that can signal the immune system to take out that cancer cell. And again, with these T and B cells, they make up a much more sophisticated immune response that has specificity to specific antigens and memory to recognize those antigens again when they're reintroduced. And the whole point of this is that, remember, when a foreign invader comes into the body, like that virus that I showed at the end of the last chapter, that infected cell has to then it has to transcribe, translate the interferon, send it out. It has to go to the next cell, and the next cell has to ramp up its defense. But by the time that happens, you get an infection and you get sick. And then it takes a while for everything to come in and clean it all up. Whereas if you have a bunch of circulating specific immune cells, they can recognize that as foreign and trap it immediately before it has a chance to ramp up, set up shop, and wreak havoc. Okay, so that's why it's so great to have this third line of defense. So we'll talk about the differences between T cells and B cells. The B cells make the antibodies. The T cells convey the message to the B cells to make the antibodies, or they will go after and kill certain pathogens. Okay, so the T cells and B cells have different functions. They also have different origins in the body, and we'll go into that as well. But they work in concert together for the overall achievement of wiping out infection. The T cell is called a T cell because it is derived in the thymus, hence T cell. Whereas B cells are derived in the bone marrow, hence the B cell. So they differentiate into their respective flavors in different tissues. But once they've done that, then they both migrate to a lymph node and then travel through the lymphatic system to monitor the body. And then this is the broad overview. This is, this is cool stuff, I think. Hopefully you think it's cool too. What's going on here? Okay. So here's an antigen. It's been introduced into the body. And now you have your phagocytic cell, your dendritic cell, or your macrophage, and it says, oh, I recognize that as being foreign. What does it use to recognize it as being an antigen? Do you guys remember? What receptor?
Is it toll-like
Yes.
receptor?
Toll-like receptor, exactly. So the toll-like receptors, they detect it and they come together, and then they internalize that antigen and they break it up into a bunch of different pieces. And then that's called antigen processing. So they take them, let's say it's a bacterial cell, they engulf the bacterial cell and they chop it up, and they take a little bitty fragment of that bacterial cell and they display it on the outside of their cell, which, you know, it's kind of like, to get a little gory, right? If you think about, I don't know, like Game of Thrones or something, where if you had an invader come into your village, what did you do? You killed him, and then you decapitated him and took off all his limbs, and then you displayed it outside your kingdom saying, hey, don't come back, right? Don't mess with us. It's kind of what these cells are doing, right? They're ripping apart this bacterial cell and then sprinkling it on the outside of its receptors to then show it to this T helper cell here, okay? It's gonna display that using different receptors. So the toll-like receptors are to bring in the antigen, and then we'll talk about the receptors they use to display it. They're called MHC receptors. They display it to the T cell. The T cell, in this case, it's a helper cell. It's going to say, hey, B cell, check out what the D cell just sent me. And then the B cell says, oh, okay, a new antigen. Let's start making some antibodies. And so the B cell will start producing antibodies specific to that antigen and then start secreting those antibodies out into the blood system that will then be circulating. And anytime that antigen tries to come back, those antibodies are ready to get it, okay? Also, the T helper cell is gonna go to the cytotoxic T cell and say, hey, we got a problem here. We've got some bacterial cells invading us, so I need you to go take them out. And so then the cytotoxic T cell will then start attacking those cells that have those antigens that the helper cell has shown it. Okay, that makes sense? Cool stuff. As I said, the MHC is standing for major histocompatibility complex. And so these are receptors that are displayed on the outside of the cell. And depending on if they're class 1 or class 2, The class 1 are used for your own cells to tell the immune cells, hey, I'm a good guy. See, I've got this MHC, it's good. My antigens should be here. The class 2 is the one that then presents these antigens that have been processed in that phagocyte. And they present them to the T cells. Okay. And so this is what it looks like. If you have a class 1 MHC molecule, it's going to be like this on the left, this green thing, and that's going to be found in all of our cells that are nucleated. I mean, basically all of our cells, except red blood cells don't have a nucleus, but it's going to signal to the immune cell that these are good cells, that they belong here. The MHC2 class is going to be found specifically on the macrophages and dendritic cells, and they're going to use this area here to present that antigen that it chewed up and is now presenting on its outside to the T cell. Okay. This is just kind of more review. B cell receptors are the ones. Yeah.
Sorry.
Yeah.
They're presenting the antigen to the T cell. They're the B cells, right? Is that what you said? You're just going a bit fast.
Sorry. So the macrophages and the dendritic cells have these MHC2 receptors, and they present the antigen using those receptors to the T cell. Okay. And then the T cell is going to go then. They can also present to the B cell directly, but oftentimes you have these T helper cells that then take it. It's kind of like, I don't know, you guys ever had a relay race with the egg, and you have to have it on a spoon, right? And you walk fastly, and you have this spoon and a hard-boiled egg, or maybe not, maybe just a regular egg. Then you have to transfer it to somebody else's spoon, right? So that spoon is the MHC. It's the receptor that's holding that egg, which is the antigen, and is going to then transfer it to the spoon of the helper cell, which is the T cell. Maybe that works. Maybe that analogy works. Maybe it doesn't. I don't know. The T cells bind free antigens and make antibodies. The T cells bind the processed antigens via the MHC class 2 of the phagocytes. Okay. And then this is just going into antigens again. Antigens are molecules that vary widely. There are, billions of them, right? Every different particle of dust may have a slightly different antigen on it. Every microbial cell of a particular species has its own unique antigens, right? So it's not just microbes. It can be other things in the environment that shouldn't be in our bodies, that our bodies can detect as being bad. Now, how do these lymphocytes, how do these T cells and B cells actually get their specificity? Well, they have a bunch of different genes that they will express or not express, or that they will rearrange the genetic sequence of them to then create essentially proteins that are complementary to the antigens. And then once they've done that, they do a clonal expansion. So the selection is they found the antigen. The expansion is now they then divide and create a bunch of different T cells and B cells with that specific target in mind for that specific antigen. Okay, so as you get older, your body has millions of different flavors. It's like, Baskin Robbins times a million. Different flavors of immune cells that specifically have one job and one job only: to recognize one particular antigen. And that's it. They don't double dip. Any questions so far? How are we doing? Is that making sense? Sorry if I'm going a little fast. I'm excited. I like it. I like this stuff. Okay. So again, T cells come from the thymus. B cells come from the bone marrow. And we said that all immune cells come from hematopoietic stem cells, which are in the bone marrow, okay? So the B cells just stay there and differentiate into the B cells, whereas the T cells have precursors that go to the thymus, turn into T cells, and then both those B cells and T cells go into the travel from the bone marrow and the thymus to the lymph nodes, where they are ready to be called out for duty. And as I said, each type of cell has a specific receptor on it in mind to a specific antigen. And some of these have receptors for self, meaning the host, the body's cells. These are going to be going around and looking at your cells and saying, okay, that's a normal, healthy cell. You can go about your way. And at some point, they encounter a cancerous cell, some kind of mutated cell. It doesn't have to be a cancer cell, I guess, but some kind of mutated cell that was still able to survive. And it says, no, you are mutated. I'm going to have to take you out. Okay. But it doesn't need to expand itself a lot, okay? It just needs a few to go around and migrate and survey the body. But all these receptors here that are designed to bind to foreign antigens, right? They are all going to go out and start expanding, meaning they're going to divide a bunch, okay? And then once they do that, they hang out. And then you have all these different varieties. And let's say you now get the reintroduction of that antigen that led to the creation of that lymphocyte. Now it says, okay, red lymphocyte, you're in. Okay, we've detected this antigen that binds to you. So now you're going to just rapidly divide and create a bunch of different clones to then take out that antigen. While these other guys, they don't have any trigger yet, so they just are poised, just hanging out, waiting in the event that that antigen that binds to them specifically will come back. Any questions so far? Thoughts, comments? Okay. A B cell makes an antibody. An antibody has a specific structure, okay? It has 4 polypeptide chains. Remember we said a peptide is just a string of amino acids, okay? Polypeptide is going to be many peptides strung together. And it's in a Y shape right here. Let's get a different, this pink's okay. Right, so it has this Y shape here. And it has 4. 1, 2, 3, 4. 2 of them are identical heavy chains. Okay, that's going to be these right here. 1, 2. 2 of them are considered the light chains. 1, 2. Okay. The light chains and the heavy chains are all tethered together by these disulfide bonds. And if you've had chemistry, you know that that's just an intermolecular force between sulfur. Okay. So these have sulfur and then they bind together, and that's what keeps this all as one package. there are different regions of the antibody. So C is going to be your constant region, means it doesn't change. It's the backbone, essentially. And then you have the V regions, or the variable regions. Let's change my color, okay? The pink stuff, that's what changes. And the change that it makes is to specifically bind to a particular antigen, all right? So this part, the constant regions from one antibody to the next, is pretty consistent. It's the little tips at the top of the Y that have the specificity, that do the mutation or the rearranging of the genome to get these variable sections that can then detect and distinguish between one antigen and another. Okay. So, lots of different genes go in to make this variable region, right? The variable region of light chains is 150 genes roughly. The heavy chains variable region is about 250 genes. And so again, they express or repress a certain number of these 150 or 250 genes to get something, to get a variable region that is complementary to the antigen. Whereas your constant regions really don't have a whole lot of genes because it's constant, right? It doesn't need to expand itself to all these different possible antigens that are out there. And we said, you know, there are billions of antigens. So you have to have a vast system to be able to get that variety that you need, okay? So macrophages and dendritic cells, they have toll-like receptors that detect the antigen and bring it in. They process it. They display it back on the outside as using the MHC class II receptors. And then how does that trigger the T cell? Well, the T cell is going to have first an antigen binding site right here, right? This is what's going to bind the antigen that's presented to it by the macrophage or dendritic cell. And then it's going to have either a bunch of CD4 receptors, which will make it a helper cell, or a bunch of CD8 cells, which makes it primarily a cytotoxic cell, okay? Yeah, just review. That's what I've all said. So CD4, helper cell, CD8, cytotoxic cells. They migrate to the lymphoid organs, right? The lymph nodes and the lymphatic system from the thymus. Here's a table. Again, you don't have to memorize these tables, but this kind of breaks down the differences between T cells and B cells, where they mature, what kind of receptors they have. So the T cell has these CD receptors and MHC1 receptors. We haven't got into the B cell really, but the receptors that they have are going to be MHC and MHC2, and they're going to secrete antibodies. Again, an antigen, that is just something that triggers an immune response. Antigenicity is what we call the property of being an antigen. Okay, so if you are an antigen, you are going to have a certain level of antigenicity. Okay, the response, the degree of the response that you elicit in these lymphocytes. You're going to be considered foreign, right? And not part of the body. What makes an antigen an antigen? Well, it has an epitope, okay? So the antigen, at the tip of the antigen is what's called the epitope. That is what's being fashioned to be complementary to the antibody, okay? So the antigen comes from the foreign invader. So again, if we use a bacterial cell as our example, the bacterial cell gets internalized and a piece of that gets displayed and shown as an antigen. And then the antigen has a part of it called the epitope that binds to the designed antibodies respective to it to then trigger the destruction of that antigen and all things associated with that antigen. Okay. Different types of antigens. There can be alloantigens. So these are the types of antigens that are within a same species, but different from one organism to the next. So blood transfusions, organ donations, those things are going to have different alloantigens between the donor and the receiving host. And as a result, the immune system will attack it and reject it if it's not adequately suppressed. Those are alloantigens. Super antigens are going to be things that primarily come from highly infectious microbes, right? So it could also be from toxins and stuff that you acquire, not from a microbe, but this isn't going to be the organ or the blood that you get. This is going to be something that is very, very harmful to you because it releases toxins. And so as a result, your body releases a bunch of toxins as well to counteract it. And it's going to release these cytokines, which are, you know, they're the communication system between the immune system to then wipe out whatever that super antigen is. Allergens, antigens that evoke allergic reactions, right? We've all heard of allergens, and if something's hyperallergenic or hypoallergenic, then it's going to either, what did I say first, hypo, it's going to not cause allergic reactions. If it's hyper, it will cause allergic reactions. And then autoantigens, molecules on self tissue, so that self cell, your own cells, for which tolerance is inadequate. So think about your autoimmune disorders. Okay, when your immune system is attacking your perfectly good cells because for whatever reason, it's getting a mixed message from them and it's thinking that they're antigenic and they need to be destroyed. Okay. Any questions on that, guys? Okay. Kind of a lot to take in, right? Like I said, immunology is complex. Has to be. Okay. A little bit more into the antigen processing and presentation. I've already gone through this, but here it is in word form so that you can kind of see it rather than just hear it coming out of me. But again, the macrophages and primarily the dendritic cells, the B cells to some extent, but mostly your macrophages and your DCs are going to be the antigen presenting cells. They're going to engulf the pathogen, express the antigen, and then trigger downstream the T cells and B cells, okay? And it's a cool process, right? So the antigen presentation has to then attract the T cells, okay? So how does it do that? It releases certain cytokines as the communication system, all right? So let's say a macrophage has this antigen. It says, okay, I've got something to show you guys. Well, no one's around. All right, I'm gonna release IL-1, and now the T cell the T helper cell is hanging out, kind of bored, you know, wishing it had something to do. And it gets a whiff of the IL-1, and so it follows it, chemotaxis, and says, ah, okay, thank you, macrophage, you have something for me to do. Once it's been given its assignment, it starts secreting IL-2 to then communicate with the B cells and the cytotoxic T cells to come on in and see what it's just been given, okay? So these cells aren't necessarily all just right next to each other. They have to send a message out, and they do that via cytokine signaling. It produces a gradient for then these cells to be attracted to. And this is a diagram of that, right? So we have, oh, not text. We have the antigen presenting cell, the macrophage or the dendritic cell. Here's the foreign microbe. It gets engulfed. It gets broken up and processed, and then the antigen is now spit out onto the MHC. That MHC then, and then the cell is going to, you know, secrete IL-1, and then now all of a sudden you have the helper T cell come, and it uses its what receptors? You guys remember? It's antigen receptors, right? To detect that antigen. What's the other receptor? What's the blue one? Anybody know what the blue receptor is? If this is a helper cell?
CD4.
Yes, all right. CD4. Just making sure you guys are still out there. The CD4 cell, because it's a helper cell, it's then going to take that antigen and now convey that message to the B cell and the cytotoxic T cell. Helper cells, cytotoxic T cells. This is a lot of text. I won't go, you know, I'm not going to read it for you, but just know that the helper T cells have CD4 receptors, and that's why they're called also CD4 cells. And what are their roles? Their roles are to communicate with the T cells and the B cells that they've just encountered an antigen from the phagocytes. Cytotoxic T cells are called CD8 cells because they have a bunch of CD8 receptors on them, which distinguish them from the CD4 helper cells. And they get activated, and then they start destroying anything with that antigen on it that is not a presenting cell, right? And how do they do that? They do that by releasing. So first they bind to the cell and kind of trap it. And then they release these things called perforins, which perforate the cell membrane of a bacterial cell, for instance. And granzymes, which are granule enzymes, then go in and break apart the cell from the inside out. Then also remember, we said that there was another type of lymphocyte called a natural killer cell that was not third line. It's second line defense, right? Because it doesn't have specificity, but it is derived from the same lineage as these T cells and these B cells. And then here's a big diagram of kind of the whole thing, right? You have the antigen from the APC, or the antigen presenting cell. It gives it to the T helper cell, or the CD4 cell, which then goes and it makes, so some of those cells will become memory cells, right? For when the antigen comes back. It doesn't need to then, it cuts out that step, right? The APC then becomes the middleman, and it doesn't need the middleman anymore. Now it can go straight to attacking that antigen. Some of the helper cells will, if they're Th2, so helper cell 2, then they will go on to activate the B cell to then start cranking out antibodies to circulate and find those antigens. If it's a TH1 or a T helper 1 cell, it's going to do slightly different things. It can stimulate macrophages to then go and continue to engulf things that are foreign, or it can stimulate and send the message to CD8 cells, the cytotoxic T cells. Once they've been in proximity to the antigen presenting cell, they will go and they'll get this boost from the helper cell, and they will go differentiate into a bunch of memory cells that will wait for the next antigen. Or they'll go and they'll start binding to that that microbial cell, or in this case, it's just an infected cell that has the virus infected inside it. And remember, when a cell is infected with virus, what does it start producing to convey to the other cells that it has been infected? You guys remember? What does it do? The innate response. Interferon gamma, or just interferon really, right? So an infected cell is going to release a bunch of interferon, which then recruits immune cells to that infected cell and sends a message to neighboring cells that it's infected so that they can start pumping out innate antiviral responses within their cells so that that virus cannot hijack its machinery and take over. It's gonna, so the T cell, it's found this infected host cell or a microbial cell, something that is expressing that antigen right here. See, remember the MHC1? It's different from the MHC2. It's not the antigen processing receptors. It's what's showing what's going on inside of it, right? So this MHC, if it's showing a mutated genome because it's a cancer cell, it's going to bind to this T cell, and it's going to detect that as being foreign. It's going to release these granzymes and these perforins that will destroy that cell. Or if it's a cell that's infected, it's going to have that viral genome in it. It's going to have the viral proteins in it, and it can take some of those that haven't assembled into a viral particle and display them on the outside of its cell. It's kind of like a red flag. Hey, I'm infected over here. Take me out. I will sacrifice myself for the good of the overall organism. Okay. Okay. Any questions on that so far, guys? We're almost done with this.
No.
Okay, good. No was an answer. Your natural killer cells, those are also from the leukocyte lineage, and they are non-specific, but they still have perforins and granzymes, which can take out other cells that are, or other cells that are infected, or other microbial cells. And then that will kill it. It'll cause it to apoptose, or programmed cell death, and then a macrophage will come in and clean it up. All right, because remember, the macrophages are kind of the vacuum cleaners that eat up all the dead debris. All right, a couple more slides on the antibodies. So there are different ways that antibodies work to trigger the death of these foreign invaders, okay? Remember, the antibodies are derived with specificity to a certain antigen. And those antigens are typically on the outside of, let's say this is a bacterial cell in this example. It's already been in the body before, so the body's waiting for it. It has antibodies circulating around, and they've detected it and they've covered it, okay? And so now one method of destruction is called opsonization, where there are so many antibodies on the surface of that microbial cell that it now can't attach, okay? And it also triggers the macrophage to then engulf it. The macrophage says, here's something that has a bunch of antibodies on it. So now I'm going to engulf it and tear it apart. You can also have a process called neutralization, particularly with viruses. If you have a bunch of antibodies that now bind to those spikes and those receptors that the virus uses to attach to the host cell, now it can't attach. It's neutralized, right? It bounces off because it's like it's inside a bubble, an antibody bubble. And so it can't reach out and hook on to the host cell receptor to mediate its entry into that cell. Agglutination is that you basically end up with a bunch of bacterial cells that are now kind of clumping together. Okay, so they can't go and infect a cell because they're attached to this other troop of cells. Okay, it's bacterial. Yeah, what's up?
Where, you know that big pack of all those water balloons?
No.
We've been throwing them.
Go.
Okay, fine.
Okay. So they can't work and they can't invade a cell because they're tethered to all these other bacterial cells because they're cross-linked via all of these antibodies. So one region right here, this part of the antibody sticks to this cell, and then the other part of that Y sticks to another cell, and they're all clumped together. Complement fixation. So we talked about complement briefly, and how it's these different proteins that work in concert to trigger an immune response. In this case, the antibodies are now going to cover the bacterial cells and bring in these other agents to then lyse it open. And then precipitation, they again kind of clump them together. These bacterial cells, for instance, they link them all together and then they're clumped together and then they just drop out. They precipitate out of circulation and then they're just eventually removed from the body. Okay. This just gives you the wording of those different approaches. A couple more slides and then we're done. We can have, when we think of immunity, we can think of it in different ways. We can think of active immunity, passive, or artificial. Active immunity, that is when the antigen is starting to, it's like the first introduction, okay? So it leads to the subsequent production of antibodies, the subsequent differentiation of memory cells, okay? And you now then have these soldiers primed and ready. Passive immunity is when you have antibodies that are passed on to an individual, okay? So it's not creating, your body isn't creating the memory. You are given that memory from something else. So think of maternal antibodies that are passed on from the mother to the fetus and the mother to the breastfeeding infant. And then there's artificial immunity, which is vaccination, immunotherapy, giving the body exogenously things that it needs to then mount this response. That's it. Okay, we made it. How you guys feel? Was that a lot? Is that kind of, was that okay? Was that digestible, or do you need to kind of marinate on that for a little bit?
I personally am going to have to marinate on it and read the book a little bit.
Okay.
Agreed.
Okay. That's fine. You know, again, like I said, this is a very elaborate system, and it's a class in and of itself, right? There are immunology courses that talk all about this, and we're talking about it in a mere few lectures. So I get that. And this is, again, just kind of scratching the surface. It gets way more complicated than this. I'm just giving you kind of the overview. And that is, I understand, can be a lot. So if you have any questions on this, shoot me an email or, you know, like in Canvas, and we'll talk about it some more. Okay. There are all kinds of good YouTube videos too that describe this and have cartoons for it and make it a little bit more interesting and visual. So feel free to reference that. Use YouTube and Google as your supplemental instructor. And yeah, if you have any questions, send them my way. Now, as you guys, oh, yep.
I'm definitely gonna have to like go into Khan Academy or something. This was just like, woo. But what specific things should we be like looking up on YouTube? Like what just because you went over all of the antibodies and B cells and T cells and natural killer cells and all the processes so quickly, I like barely have time to scroll anything down. What can you like maybe run through the slides and just, this is a process you should know?
Yeah,
you know?
so I think what you can do, if you want to look up things like antibody production or antigen presentation or just T cells and B cells. I mean, there are, Crash Course is a good website. It's got the guy who talks even faster than I do explaining things, but they have, you know, like cartoons and stuff that go along with it. Really, in a nutshell, I want you to know the difference between first line, second line, and third line, which you guys do. But then when we get into the third line, what's kind of the process? All right. Well, you have second line cells like your macrophages and your dendritic cells. What's step one? Step one is they have to encounter the antigen. Step two, they process the antigen. Step three, they present the antigen. Okay. So in those three steps, there are certain characteristics you need to know. Like one, what kind of cell is it? Well, it's going to be a macrophage or a dendritic cell. Some cases a B cell. But primarily, I'm concerned with you guys knowing that it's dendritic cells and macrophages that are second line of defense. What do they use to detect that antigen? They use the toll-like receptors. They internalize that antigen and they cut it up and they spit out the little pieces onto their MHC class 2 receptors. And from there, the T cells come. Now, what do you need to know about a T cell? Why is it a T cell? Because it matures in the thymus. What receptors does it have? CD4, which makes it a helper cell, or CD8, which makes it a cytotoxic cell. What does a CD4 cell do? It takes that presentation from the macrophage and shows it to the B cell to start making antibodies. The cytotoxic T cell then goes out and attacks those things that have that foreign antigen on them by using perforin and granzyme. Okay. So there's a lot of slides, there's a lot of things we covered, but there's really kind of an overarching process that hopefully can rise up out of all of those slides, and that is what I kind of outlined. What do you need to know about an antibody? Well, they come from B cells. B cells mature in the bone marrow. They have heavy and light chains. They're strung together by disulfide bonds. They have a constant region that doesn't change and a variable region that does change. Do you need to know that the light chain has about 150 genes and the heavy chain variable region has about 250 genes? No. I want basic concepts, the overall process. That's more important to me. Make sense? So if you go to Khan Academy or Crash Course or other YouTube videos and you see the same message kind of over and over again, or if you see something that's talked about in all like a common denominator, that's probably what you should be focusing on. Okay. Any other questions on this?
I have a question.
Yeah.
The natural killer cells, so those are considered second line defense because they're not specific?
Correct. So they do come from the same precursor as T cells and B cells, but they don't form a memory and a specificity to individual antigens. So then they aren't in the third line, they're considered second line because they operate on an innate level like macrophages and dendritic cells.
Okay, so then do killer cells have toll-like receptors or some sort of receptors or that we need to know about, or are they different?
No, don't worry about that. Just know where they come from, right? What lineage they come from. Know that they don't come from monocytes, and know that they aren't adaptive. So they're second line. And I mean, you know, if you know that, but because they come from the same lineage, their method of attack is perforin and granzyme, like the cytotoxic T cells, which should make sense because they're from the same lineage, right? So they have the same expression of certain genes. And so therefore their mechanism is going to be along a similar line because they have a more close common ancestor, if you will, right? So that kind of thing is what I'm looking for.
Okay.
Great questions. Any other questions? Maybe sometimes it's, if you feel overwhelmed, it's hard to think of questions, but if they come as you're reviewing this stuff, let me know. Now your topic is due today, and I've had some questions on that. Like, I don't really understand exactly. I've looked at the video, but I'm not quite sure what you're looking for. All I'm looking for is for you to pick something related to microbiology and research that. This is you as a grad student or you as a scientist, and you want to get funding or approval to do a certain project. So that's going to involve one type of microbe, right? It's not going to be, I want to look at bacteria and fungi and viruses. No, you say, like, I want to look at this specific virus. I want to look at SARS-CoV-2. Or maybe it's a disease. I want to look at UTIs. What causes them? What treatments are there? What treatments are insufficient? Are there any new treatments that can be discerned? You know, that kind of thing. That's what I'm looking for with your topic. Now, your topic doesn't have to be anything super detailed at this point, right? Once we get to your outline, then you need to have more detail. But right now, you just need to narrow it down to one aspect that you would like to research. And I encourage you guys to pick things that interest you, because it makes the process more enjoyable. So try and think about like, obviously SARS-CoV-2 is a obvious one because we are in the throes of that. But let's say you had a particular bout with a nasty infection at some point, or you have an autoimmune disorder. Like, you don't have to, like, that's your privacy, that's your personal, you don't have to, certainly don't tell us why you're choosing a topic if you don't want to. But maybe you had a family member or a friend that had some kind of trouble with a microbe, and then that makes it a little bit more, it sends it home more, okay? And if you don't, I mean, if you're lucky that you're like, man, I don't know, I've been pretty healthy my whole life and my family, I can't think of anything that's micro, then start looking at the news. What's going on? Like, just recently I mentioned the brain-eating amoeba. There is the Eastern equine encephalitis virus. It seems to come out every year in the northern Midwestern states, but it's back again, and there's been several people infected with that. There's all kind, like, with global climate change, Now we see the temperatures rising, and microbes that were once restricted to certain regions are now becoming, expanding their territory. Maybe you want to look at it from an environmental aspect. Maybe you don't want to do infectious disease at all. Maybe you say, hey, I want to find bacteria that will break down plastic, because the ocean is chock full, and they're saying that pretty soon there'll be more plastic weight than all the fish in the ocean, right? And that's a problem. So let's find some bacteria that can provide a service for us. Or maybe you want to work with algae, to get natural biodiesel or some kind of natural alternative energy source. It's, it really is wide open to you guys. I just want you to pick a topic to focus on. And it's not this isn't a research project like many of you have probably already had, where you just give an overview of what's going on in that area. Maybe that's the confusing thing, right? When I say research proposal, this is going to be something that you are going to do as a researcher, okay? I don't want you to just summarize what's going on in that field. I want you to figure out what's going on in that field, and then figure out how you are going to take it one step further. And the great thing is, you don't have to take it further. It's all conceptual at this point. But it trains you guys to think like a scientist. It trains you guys to read like a scientist. And hopefully that will make your whole life, different in a good way. You'll be more critical of information that comes in, because we're all bombarded with lots of information right now. And a lot of it is misleading. A lot of it is just headlines that we look at, and then we form our opinions based on the headlines and don't actually read the article. And so this process, because this, for LTCC, this is an upper level course. This is BIO210. So the expectations are that you've been trained in how to use the scientific method. And so that's why I'm having you guys do this. Any questions on the research proposal or on the material that I covered so very quickly?
I have a question.
Huh?
So I have a general idea of the topic I want to do. Do you want us to also have in mind how we want to further the research that we found at this point, or no?
If you've got that, then put it in there, yes. But all I'm doing now is forcing you guys to narrow down to a particular thing.
Awesome.
Okay.
Awesome. So, yeah, because it's going to take me a little time to work through some of this to really figure out how I would further the research, but I know the broad topic is interesting.
Absolutely. And so the outline is not due for another 2 weeks. Okay. So today is designed to get your topic established. And then the next 2 weeks is designed for you to be thinking about that topic and where you want to take that topic. And then after that, you think about, well, what experiments do I need to perform and conduct to get it to that point? So it's a snowballing effect. This is the little tiny clump of snow that we're just putting together. We're going to make it bigger as the quarter goes on. Okay. Now, if you just say, I want to look at the microbiome, that's kind of vague, right? Maybe say, I want to look at the microbiome, the gut microbiome. Okay, that's a little bit more specific. I want to look at how the gut microbiome affects humans psychologically, because there have been studies to show that the gut microbiome communicates with the brain and can lead to things like stress and depression. Or people with PTSD can have some type of alleviation by manipulating their gut microbiome. So now we're getting into a little bit more detail, but you don't need to know what to do with that. You need to know that that's the topic I want to look at. I want to look at the effects of the gut microbiome on brain chemistry. That's good enough. And then in a couple weeks, you say, well, I want to look at the effects of the gut microbiome on brain chemistry by using a mouse model and taking germ-free mice and then colonizing their gut with this type of microbiome and then subjecting them to stress and seeing how they handle that. And then this other set of mice are going to be given a different microbiome and see how that, or maybe you want to do a human trial, that's fine too, right? But then you get a little bit more granular as time goes on.
Carla, I do have a question. So out of the three that I was mentioning to you of what I'm going to be looking at with the repurposing of drugs,
Yeah.
would it be okay for now, since I haven't, like, I can't find more info, to choose like one, like say if I want to do fungi, and then later on, once I do more of my outline for in 2 weeks, I can add to that. But for the meantime, saying I'm going to be focusing on like repurposing of drugs for the treatment of fungal infections. Would that be sufficient for now?
Yes. Because you've broken it down into one particular microbe and what facet of that microbe, treatment, and what type of treatment? Well, novel treatment. Through repurposing, but still it's a novel therapy. So yeah, I think that's plenty at this point. This process for today is not to get you guys stressed out, but to get you guys committing.
I have a question.
Mm-hmm. Yeah.
Is there any possible way to link leukemia to microbiology? Because I know it kind of fits in with the immunology section, but I don't know if it's possible if I can do a topic about it.
Yes, you can. You can. Who was that?
Really, it's a DNA mutation that happens.
Yeah.
It's not really a bacteria.
Who am I talking to?
Janae.
Ah, there you are. Okay. Sorry. Yes. So what did we just talk about this week? We talked about the immune system. So, if we cover a topic in this class, it should be fair game. And, leukemia is a cancer, but it's a cancer of the immune system. Mm-hmm. Right. And so if you guys want to deal with some kind of immune disorder that's not exactly linked to a microbe, then that is fine with me. You could look at the use of immunotherapy in the treatment of leukemia.
Okay.
So another student asked about melanoma. There's not a whole lot of information that I know about of microbe-associated melanoma per se, but the gut microbe, the gut microbiome can trigger certain immune downstream consequences that will either effectively go after melanoma or not. And melanoma is a really great candidate for So going back to immunotherapy, there's been all kinds of FDA approvals of the use of immunotherapy using antibodies to treat melanoma. And your microbiome can also lead to the expression of this thing called PD-L1, which is the communicating signal that we won't talk about in this class, but it allows T cells to recognize cancer cells and destroy them.
Okay.
So yes, if it has to do with the immune system, I'm fine with it not being exactly linked to a particular microbe.
Okay. I guess maybe could I email your Canvas inbox you ideas to make sure it's makes sense?
Yeah.
Okay.
Absolutely.
Okay. Because I'm always trying to figure out like exactly what's happening and how it works, and I feel like that would just be the easiest topic for me and most interesting. So. That's
what I want. I want you to pick something that's interesting to you. And if you really have to dig in and find out how something works, then that's great. This is a project that is designed for you.
Okay, cool. Thank you.
Mm-hmm. Any other questions? No? Okay. Well, you have till the end of the day to come up with, to submit a topic. If you feel like you're just struggling and you just can't find something, just pick something. And I won't hold you to it if you can come up with something in the next couple days. Okay. But I need you to submit something for this so that you get credit for it. So if you're struggling between a couple different topics, you are welcome to just eeny, meeny, miny, moe it and submit that one. And then over the weekend, if you think about it more and like, actually, I should have picked that other one, just send me a message on Monday and say, hey, I'd actually like to change it to the other one, and that's fine. Okay. I want you guys to have enough time to progress the topic. So, okay. All right. Well, unless there's anything else, you guys have a good weekend, and I'll see you on Monday. We'll start talking about viruses, and we also have the Tuskegee Syphilis Project. So read over that. There's no summary that you'll be writing. You'll just be answering those questions at the end of it, and then we'll talk about it. Okay. All right. Well, thank you guys for joining. Go take a break. If you don't have anything next, get some fresh air. I guess I haven't really been outside. I don't know if it's smoky or if it's a nice clear day, but yeah, un poco. Yeah. So do what you need to do to decompress, get your mind cleared, and then think about your topic if you haven't already submitted that. All right. Okay. Take care, everybody.